So this part of the lecture is about prions. And prions are really interesting. They are infectious proteins. So there's no genetic material, there's no capsid, there's no cell. These are actually normal proteins that are encoded in an animal, in an organism. For instance, on chromosome 20 in humans, you encode this normal prion protein. And when it forms properly, it works just fine. The diseased prion, okay, is just misfolded. So it's the same sequence, it's not a mutation, it's just misfolded. And what's really interesting is all of that you learned about proteins and how you can denature them, and you can put them in acid, or you can change the salt, or you can raise the temperature. This doesn't work for prions. So let's talk about these. Again, prions, normal proteins that for some reason misfold. Once they're misfolded, they trigger the other normally folded prions right here to misfold. So it triggers normal, proteins to misfold. And it's like a domino effect. So they keep misfolding and misfolding, and what happens is, you can see, instead of being separate proteins over here in yellow, they start to aggregate or clump up. And what happens is then they start to glob up the cell, and eventually the cell dies. Prions are infectious because you can get them from another source. And so we heard about mad cow disease. So those are prions in cattle. And what happens is they contaminate the food or the meat you eat. Okay, so the prions are going to infect neuronal cells, but when they slaughter the cattle, some of this brain and nerve cell contaminates the meat. You can try to cook it, and it still does not denature the prions. The only way to get rid of prions is incineration, burn them up. So what's happening is, as you eat the burger, the prions are absorbed into your gut, and they are stable. They do not get denatured by that acidic pH in your stomach. They can travel then eventually to your brain and cause damage, cause that misfolding. So here's a couple of pictures of brain cells, and what you're trying to see is these big empty spaces. Okay. So the brain becomes spongy. So BSE is bovine spongiform encephalitis. Okay. Kuru is a prion disease due to cannibalism. So in some small tribes, when a member of the tribe would die, they would eat the brains. And what they found is sometimes these people died because of Prion disease, and then the people who ate the brains basically ingested the prions and then also came to die. Scrapie is in sheep, so you may have also heard of chronic wasting disease in elk and deer. Creutzfeldt-Jakob is actually a genetic form in humans. So sometimes there is a mutation that causes the prion. This is very rare. What's also interesting is these misfolded proteins do not cause an immune response, so your body is not fighting it off. And sometimes this can take like 35 years to manifest into brain and nervous system damage that eventually kills you. They're looking at this idea of misfolded proteins in other nerve diseases, right? So Alzheimer's, Parkinson's, not mad cow, Alzheimer's, Parkinson's. There's other naturally expressed proteins that seem to be able to misfold and cause that aggregation and thus destruction of brain and nerve cells. So, prions are infectious proteins, cannot be denatured. You have to incinerate in order to get rid of the protein. They're infectious because they can go from organism to organism. So the final slide I want to show you as we wrap up this section is just the relative sizes of the pathogens we've talked about. So we've previously talked about bacteria. So here this green thing is a big bacterial cell. Okay, behind it is supposed to be a human eukaryotic cell. There's a bunch of viruses in there. Here's a ribosome. So the prion would be even smaller, right? Because it's just a protein. All right, that's it for the first set of pathogens. Talk to you again soon.